Bio 208. This is Dr. Grenier. Again, we are going to be looking at this page in our lab manual. And why does it say spotlight video? I'm trying to spotlight me. I don't understand why it's not on me. I never know why nothing works, but hopefully it'll magically just continue to happen. Okay. So I do need to real quickly on the participants list, I need to quickly download all your names because otherwise it won't let me find your names later. So that way I can do attendance. And for anyone that comes in late or anyone that's watching this recording, make sure you email me so I know attendance that you did attend / watch the video to be counted present in lab for week 4. Okay, so in chapter 21, we're going to start with blood vessels and the histology. Okay, and so since hopefully you can follow along, I'm going to try to spotlight more on the model. And then I have this secondary model that we can also look at. This one is not usually on your test, but it's a good model because it does show some of the features. Can y'all see this okay? Y'all can move to this table if you really want to see a little better. Okay. All right. So histological layers. In the heart, we call them endocardium, myocardium, epicardium. Okay. So in the vessels, we're going to call them tunics. So we're going to have a tunica intima, like your intimates. I always think of like, you know, the ladies department, they have the intimates. So that's where you get the bras and the panties. So the tunica intima, the tunica media, which is going to be the middle, and it's going to be the muscle-rich layer. And then mute me. Y'all are Who is that? Mallory, mute me. Mute your phone, mute your microphone. It won't let me mute you, I think. Or let me find you. Okay, you're muted. Okay. And then the external layer is going to be the tunica externa. Okay. Now, as it shows in the table, as it shows the table from the book and the picture, the material in your lab, each histological layer is going to have potentially 2 to 3 tissue types present. Okay, so for the tunica intima layer, and again for the tunica intima layer and for the first of these vessels, we're going to be looking at our biggest vessels, which are our elastic vessels. Okay, so the tunica intima layer is going to be the innermost specific tissue is simple squamous epithelium. And if you remember from the heart, those are known as endothelial cells. So they are flat and squashed, right? And you can see them as this thing jumps out at me. You can see them here as these cells, okay? Those are the simple squamous epithelium, also known as endothelial cells. And if you go back to chapter 19, to the clotting process, remember, these are some of the cells that can release growth factors, release signaling molecules to downstream areas, to inner areas, to tell the smooth muscle to constrict or relax. All right. And these are also tissues that can communicate to each other and try to grow and multiply until they touch again during the process of the clot going away. Okay. So in this image or in here, I think this is number 7, and it's this orange cell looking layer that is going to be your tunica intima's innermost cells, which is your simple squamous epithelium or your endothelial cells. Okay. Now, as the book talks about it, in our arteries, because there's so much muscle, all right, the tunica intima is going to look more like a circle or a nice like rounded hole, all right? But in our veins, where there's less muscle and there's less kind of oomph to the veins, And again, it's in the slide here, if you can see it. Veins are going to be, again, like these crazy oval, long shapes, because they're going to kind of try to form and take a shape kind of based upon the forces and the pressures around them. Okay, so the tunica intima may look like it's a nice circle in an artery on the inside, and then in a vein, it's just going to be that lining for this kind of oblong, long, crazy look. Okay, now for every epithelial tissue, there's normally connective tissue right behind it. And that connective tissue is going to typically be, somebody mute me again, okay, is going to be areolar connective tissue. Okay, so right behind, and it should be this little gray, all right, that should be your areolar connective tissue. So that's where you're going to find the capillaries that are going to feed the endothelial cells some of their nutrients like oxygen and glucose and amino acids, because they can't get it from the blood. Okay, that is where you'll find some collagen and elastin and material. You'll find fibroblasts or fibrocytes, which lay down the proteins of the areolar tissue. You find some random, maybe white blood cells, some random, maybe adipocytes, and anything that can live in connective tissue. The fluid in this gray area is going to be known as interstitial or extracellular fluid. Okay? All right. Now, for our elastic arteries, okay, and our elastic artery in this model is right here. It's going to have this white meshwork layer that is going to be, if we go back again to your book and you go back to the table, this is going to be an internal elastic membrane. It is going to be an elastin-rich layer that kind of begins to be where the areolar tissue kind of ends and more elastin grows, and then right before you start your muscle layer. Now, this model is inappropriate in that it shows this elastic layer in the veins. You do not have an internal elastic membrane in the veins. So if I was grading this model, that would be a big X for wrong. Okay, so in the artery, you have this internal elastic membrane. It's technically part of the tunica intima, but it's kind of like the fence where it represents where the tunica intima ends, and it represents where the tunica media begins. Okay, and it's elastin rich, so that way if this is the aorta or this is some of the big vessels and they get, again, like a big bolus, a big kind of marble-like push of blood as a big entity, they can kind of stretch and accommodate that and then come back to a normal size and shape. Okay, now the tunica media layer is going to be the muscle-rich layer, and the muscles are going to tend to be arranged around, and when they're around, when they contract, if somebody's in the waiting room, you'll let them in, all right? And if they are going to contract, they're going to constrict the vessel, so the lumen or the radius is going to get smaller. Okay. Somebody's getting my attention, but anyway, all right, the lumen or the radius is going to get smaller. When they dilate, the lumen or the radius is going to get bigger. Okay, these are smooth muscle cells. So remember, smooth muscle cells can be told to contract by the nervous system, like norepinephrine and epinephrine. They can be told to contract by angiotensin 2, by ADH, because we learned about that, and that's going to affect afterload. They can also be told, like we were looking at with the vascular spasm, to be contracted by things like histamine, prostaglandin, adenosine, potassium, things that are telling them damage has occurred. All right? On the flip side, these smooth muscles do also know to relax or be told to relax. Typically, that's by nitric oxide. That's why people take nitrates or sometimes put something in their mouth when they're having a heart attack to try to dilate and decrease that afterload. Okay, and there's a few other, again, relaxating materials out there, but for the most part, we're really good at telling smooth muscles to constrict a little bit more not necessarily as good at telling them to relax. Okay, especially when it comes to altering blood pressure and cardiac output. Okay, all right. Now, moving into between these smooth muscle cells, there will be a little bit of areolar connective tissue because these smooth muscle cells do need room for capillaries to come in and provide them with nutrients. And again, as the muscle cells are going to squeeze and contract, they're going to be pulling on some of the connective tissue in front and behind them and a little bit in between. Okay. The next layer is going to be our tunica externa layer. All right. And the tunica externa layer, like the tunica intima layer for those elastic arteries, is going to start with an external elastic membrane. Okay. And that external elastic membrane is another kind of white meshwork of spiderwebby material here that you see. All right. And it then bleeds into a collagen, dense, irregular connective tissue that makes a capsule on the outside. Okay. Now, on the veins, you're going to see that the tunica externa is going to be a little bit thicker, and it might even completely overpower the tunica media layer. And as you go on the veins to smaller and smaller and smaller veins, the media layer is going to be the one to disappear first. Okay. On the artery side, on the arterioles, the external layer will actually disappear and the muscle layer will get smaller. So when we get to those systemic arterioles, they are going to have 1 or 2 layers of the smooth muscle cells and the tunica intima layer. And that way they're still really small, but they have the ability to still, as a small vessel with smooth muscle, constrict or dilate themselves. Okay, now back to our tunica externa layer. In the veins, and you can kind of see it right here, you might see in the veins fat is heavily deposited in there. And that's part of, again, why some of our tunica externa layers kind of have this ability where they can stretch a little bit, but they can't really stretch beyond too much because, again, fat does not have a lot of give. Okay. The other thing you might see with the tunica externa layer in veins is that they will randomly start to have smooth muscle cells embedded in them. All right. Now, the smooth muscle cells that end up in the tunica externa layer, they offer the ability to a very small degree that when the muscle layer for the vein disappears, there's still a little bit of muscle randomly in the tunica externa layer, so it might be able to tweak a little bit of the vessel, but not really to the same degree as if it had a muscle layer. Okay? All right. These tunica externa layers, because just in our picture, veins and arteries, especially our big ones, tend to run together. The collagen of this tunica externa layer and the collagen here, they are going to, again, kind of meshwork and weave so the two vessels can run together. And as they run together, then it's less likely for this guy to get kinked or pinched off because you're running with these two vessels. And in many cases, the nerve is running and their epineurum has collagen in it and it's running with them. So it'd be like running as a tube, 3 tubes together instead of one garden hose that can get twisted and kinked up. Okay, so keep doing that. So if we look at this again in this model, all right, tunica intima is this layer here and the directly underlying connective tissue. If this was a major artery, an elastic artery, we would then also expect an elastic internal membrane. Okay, the tunica media is the middle. The tunica externa is out here. And again, if it was a major elastic vessel, there would be an external elastic membrane. Now, in the areolar connective tissue of the tunica intima, again, there can be adipocytes deposited there. But in general, we don't like adipocytes to get deposited there because as they store fat, adipocytes are cells that are not known to be stretchy and malleable. one of the things we learn is that when you have damage by high velocity, think about like cars on roads running really, really fast, the road gets torn up. So high velocity blood is tearing these cells apart a little bit, okay? High pressure, because it's a vessel close to the heart, seeing that high pressure of the blood pushing on it, causing friction on these cells, is going to tear them apart. So one of the things we know endothelial cells can do when they're under high pressure, high velocity, is they will start to put on their membrane a receptor for a protein known as the LDL protein. The LDL protein, if you don't remember, is the low lipodensity protein. It is a protein that wraps around triglycerides and fatty acids and cholesterol so they can travel in the blood because those molecules are hydrophobic. Well, when the endothelial cells bind their receptors to more LDL proteins, the LDL proteins, instead of taking that fat to cells that want to use it for energy, they take that fat and they deposit it in cells and in the external environment behind the endothelial cell. And if you think about it, in the short term, the endothelial cell that's damaged, if it has a little bit of a fat layer behind it, that's going to push the blood and the water of the blood to stay in the blood, right? And so in the short term, it's an okay little backup to say, I'm putting a tarp behind me so the water stays in the tube. The problem is once you turn these endothelial cells on to have higher receptors numbered wise and collect the LDL proteins, you can't turn them off. And so what starts off as a little bit of an assistance eventually gets to a place where the deposit starts to grow into a large grouping, and the elastic forces will accommodate as much as possible, but you're going to get to a point where, can this vessel go any bigger? No. If it goes any bigger, it's going to burst. So what then happens is you get to a point where it has no choice but to continue to take the fat and grow and deposit and push inward. And that's when we talk about people get stents because they've occluded the vessel in their coronaries. Again, through natural circumstances, the minute you start breathing, the minute you start letting blood in your vessels, you're having friction on some of your major elastic arteries. And every one of us hopefully is in this stage of atherosclerosis. But if we live long enough, chances are most of us are going to get to some over here. All right. And so eventually we will potentially need stents to reopen those arteries and keep them open. Okay, so part of the reason why a heart healthy diet is normally low in fat and cholesterol is if you are turning these cells into LDL receptors or in high density, you don't want them to have a lot of fat and LDL proteins carrying fat in your blood. So if you can, as a heart disease person, eat less fat, have less cholesterol in your system, LDL isn't bad, but high numbers of LDL proteins and high numbers of these LDL receptors means there's greater incidence that the fat is going into the areolar connective tissue of the tunica intima and not to use elsewhere. And that's why it's bad. It's really not bad, but that's why we call it bad. Okay? All right, so those are our vessel histology. Okay, and again, as we go through it, the major elastic arteries are going to have those internal and external elastic membranes. Veins do not have it. Veins, we're going to see again the smooth muscle cells randomly in their tunica externa layer. Right? As we go to muscular arteries and smaller veins, on the vein side, the tunica media goes away. On the artery side, you're going to see those internal elastic and external elastic membranes go away, and the muscle becomes important. Then when you go to the arterioles, there's no more tunica externa. You're down to 1 to 2 layers of contractile smooth muscle cells and the intima. On the vein side, a very small amount of tunica externa, maybe a few random smooth muscle cells in there, and the tunica intima. Capillaries, it's only the endothelial cells lining, and then the external interstitial extracellular space is going to be the same interstitial extracellular space for the cells of that organ. Okay? All right. Questions? Okay. You guys can look at these if you would like to get a little bit better view. Questions from anybody in our group here? Is there something in the chat? So there should not be any internal elastic membrane in your veins, nor should there be an external elastic membrane in your veins. Okay, those are something that you should expect to see in the elastic arteries. Correct. Any other comments or questions? Okay, so while I have you guys here, let's go back through some of our major blood vessels, and let's review the ones we should all hopefully know on our heart. Okay, so everybody remembers that this is the right atrium, okay, or right atria, and it is fed by this major vessel, which is going to be our superior vena cava, and then the one coming here is our inferior vena cava. Backside, if I can get this off. Oh my God. I am smarter than the model. There we go. Backside, remember that this is the coronary sulcus. So the right atrium is fed by the inferior vena cava, superior vena cava, and the coronary sulcus. Okay? Now, the heart is fed, again, coming off the aorta to the right. This is the right coronary artery, and it's going to whip around under the right auricle in this coronary sulcus, and there will be a right marginal artery that comes down at some point, and there'll be smaller branches that come down, and then the right's going to whip around and become the posterior interventricular coronary artery. Okay? Now, the left, which comes off over here, the minute it hits this kind of surface place, it's going to fork. And when it forks, the vessel that goes down into the anterior interventricular sulcus is going to be our anterior interventricular coronary artery, also known as the left descending artery. Those are its appropriate names. Now, it has a nickname as the Widowmaker, but you're not supposed to know nicknames. You're supposed to know real names. Okay? The vessel that whips around under the left auricle, all right, that's your circumflex artery. These are the two big arteries that get a lot of attention for stents, especially this one, because the blood going in here is under a lot of pressure because it gets pressure from, it's coming straight from the left ventricle and it has high velocity. And then all of these, again, contract and relax. The vessel is constantly being squeezed, okay? And anything that's squeezed really, really extreme and then stretched really, really extreme, those endothelial cells are going to get damaged. So we're going to see that atherosclerosis process in high amounts, even in healthy people. So all you youngins who think you're healthy, you still have a little bit of that fat LDL deposits. And that's why whenever you get blood work and they say, oh, your LDLs are good, part of what they're saying is the incidence, the statistics that you're taking those LDL proteins, and instead of depositing them in the liver and adipocytes and using the fat, you would be depositing in these vessel walls, making these vessels less able to be stretched and less able to contract. Because once the fat gets in there, the vessel doesn't move as easily because fat doesn't move. Okay? Now, the left marginal comes off the circumflex, and then that's how most of our left ventricle is fed. For the veins, in the anterior interventricular sulcus, this is the great cardiac vein, and you have to say great cardiac, you know, put those two words together. The one back here in the posterior is the middle cardiac vein, and then you have your small cardiac vein that normally runs with the right coronary, but this model doesn't show it. Okay? For the pulmonary, and again, anytime we talk about pulmonary, you have to specifically say pulmonary. the big vessel coming off the right ventricle, this is the trunk because it's one vessel. Then the minute it splits, these are going to go to the left lung. So these are the left pulmonary arteries. And then the ones back here going to the right lung or the right pulmonary arteries. And then they split further and further and further. The ones coming back, and it shows 2 from the right and 2 from the left, those are your left pulmonary veins. And remember, pulmonary circulation, oxygen-rich blood is coming in the veins to the left side of the heart. Deoxygen, or the low oxygenated blood, is leaving the heart in the arteries. And that is the big question with why, if you ever see all arteries carry oxygen-rich blood, you always say false, because pulmonary arteries do not carry oxygen-rich blood. Okay, so that's why it's always, when they say that always, that arteries always, no, pulmonaries are the exception. Okay, all right, so again, coming off the aorta here, the first vessel, and this is beginning to be the page in your lab manual. Okay, so coming in this page, first vessel is your brachial cephalic artery. And then what I like about this model, all right, is when you show that coming off the aorta, this first, it very quickly branches into what will become your right common carotid and then your right brachial artery, you know, or going to the right arm, okay, or the right subclavian, sorry. Okay, and then here's your left common carotid, and then here's your left subclavian artery, right? For your veins, you can see the 2 jugulars are coming down, and then your 2 subclavians. And we're going to learn about lymphatics, this little fork right here between the jugular and the right subclavian and the jugular and the left subclavian. That's where your lymphatic fluid ends up back in the system. Okay? All right, so those are some of the major vessels on your heart you have to know. All right, now we're going to jump to this. Okay, so I guess here is our aorta as it continues down. I don't know if y'all can see that all that well. Let's see if I can get you a little closer. So here's the aorta that's coming down. All right, now we don't have great pulls off of the aorta here, but note that part of the carotid and then part of going into the subclavian, there is a little vessel. You can't even see my finger, and it's in one of the pictures that comes here. That's sometimes known as your vertebral artery, and it's in this picture here. You can usually see it right there, kind of going towards the vertebra and the neck, right? So your carotid's here, and it's a little bit more superficial. Okay. And then you'll have the internal thoracic and the thyrocervical trunk. And then the next big artery is the one going into your arm. That is going to be your axillary, and it's the one going into your armpit. And eventually your axillary is what we're going to call the brachial artery when it's by your tricep and your bicep. And then it forks at your elbow. And remember, it's going to get the name from the bones. And so the radial and the ulnar artery, all right, and which in anatomical position, which bone is more lateral? We remember? Should be the radius, right? So the radius, remember, is going to go towards the thumb on the lateral side. The ulna is going to be the one going towards the pinky, all right? And then you'll have superficial and deep palmar arches, and then you'll get it into the digits, this picture, okay? All right, so the good thing about the arteries is armpit, arm, and then bone names. So they kind of get their names from the anatomical location and the major bone or feature. All right, for this page, after you get to the common carotid, there is a connection where the right and left form this circle, and that's part of the reason why someone could potentially have problems in one carotid artery, but their brain still works. It's called the circle of Willis. All right, I'm not going to ask you anything other than the common carotid and the right or the internal, external circle of Willis. That's about as far as I go. Okay, so you don't have to keep going up higher. If you do go higher, it's because you're going to work for a neuro person and you're going to be playing in people's brains. Okay, now you'll have to do the picture because our model doesn't quite show it for things that come off the aorta in the thoracic area. All right, and they kind of get their names again. The one that's going to some of the major bronchi, or it's going to be called the bronchial artery. The one that's going to go to the pericardial sac and feed the connective tissue and the mesothelium, that's your pericardial artery. The one that has to go and feed Mr. Esophagus, which is running with the trachea, then with the aorta, all right, that is going to be the esophageal vessel. Then you have some arteries that need to go to these, again, muscles between the intercostals and the periosteum of the bone around the ribs. That is your intercostal. And then remember, the phrenic nerve controls the diaphragm. So you're going to need some blood vessel to go to this area of the diaphragm, and that's going to be a superior phrenic vessel. Okay, so the vessels are being named for what they're going to feed in the thoracic region. All right. When we get into the abdominal, and this does kind of match up, so let me try to get this a little closer. Okay. All right. We go through the esophageal hiatus. Okay. And then we have these three vessels. I don't know if you can see it all that well, but you have these three vessels that come together and out of the aorta kind of as a unit. That's known as the sciatic trunk, okay? And the one that's going to go to the stomach is going to be the gastric artery. The one that's going to the spleen is going to be the splenitic. And the other one that's going to go towards the liver is your common hepatic. And what's cool about the liver is this vessel is bringing the liver 1/3 of the blood it gets. The other 2/3 is coming from the hepatic portal vein. So this is the only vessel bringing oxygen-rich blood to the liver. So the liver gets 2/3 of its blood from the digestive system that's already deoxygenated, and only 1/3 of its blood from arteries that bring oxygen-rich. And again, the liver is doing a lot of things with metabolism, with the digestive tract, and that's part of the reason why it wants all that blood from the digestive system through the veins. Okay? Okay. You'll see these, so this would be your superior mesenteric is this nice big vessel here. The one going to the kidney is the right renal and the left renal. And remember, no other artery is bringing blood into the kidneys. So this is why, going back to chapter 19, this is why the kidney makes the EPO, because it's going to get 500 milliliters of blood, and if 500 milliliters of blood is not enough to give oxygen and glucose and fuel and everything to the cells in the kidney, there's no back door. There's no other vessel to bring blood into. So the kidneys then would tell the blood to make more red blood cells by releasing EPO. Okay? And then you have the gonads. So these little vessels coming off are going to go normally to the ovaries. And then you have the testicular parts. You have the one coming down right here. That's your inferior mesenteric, right? And then your split, you will call the right and left common iliacs. Okay? And then the right and left common iliacs, when you go to this page, you'll see will become an internal and an external iliac. The internal is going to go to things around your pelvic floor. And then your main artery in the leg, of course, everyone knows is our femoral. And then our femoral will run into the back of the knee. So it'll start to be called our popliteal, again, by direction, that anatomical term. And then it's going to split. And one of the parts going to go towards the front. That's why it's the anterior tibial. The other one stays on the back side for the posterior tibial. And then you start getting into the anterior tibial is going to go to help, again, the flexors lift the toes. That's why it's dorsipedal. Whereas the posterior tibial is going towards the back to help point the toes. So it's the plantar. And then they go through the arches and the toes. Okay? All right. Most of the time for the veins, different names. So you have to just memorize them. So again, the carotids are going to be lined up and paired with jugulars. All right. There are some cases where the artery and the vein are named the same. So the subclavian arteries, there's also subclavian veins. But for the most part, the veins have weird or interesting names. Okay. All right, and then some of the veins of the arms, there is a radial, there is an ulna, there is a brachial, there's an axillary. And again, we have cephalic, medial, antebrachial, basilic. And just an FYI on a clue, if I'm going to ask you the radial artery, I'm probably not going to ask you the radial vein because that's like a gimme. I'm not going to ask you the same basically named vessel. So if you think of something's the radial artery, then chances are I will not ask you the radial vein. I will try to get you, again, breadth of different vessels to make sure you demonstrate to me you know this material. Okay? These you'll have to look, and because they're in color, for the veins, there's some light blue versus dark blue that tell you, like, I think the light blue are the ones more superficial and the dark blue are the ones more behind. Follow your book to be able to label them. And the same goes with the leg veins. There's no femoral vein. Remember, it's a great saphenous and then the small saphenous. And what's important about those saphenous veins is if we were to decide in the heart that we need to put a piece of vessel to replace our anterior interventricular coronary artery, sometimes we'll take one of the small saphenous vein or some of the mammillary veins, and we'll put that piece of vessel to do the bypass. And what's interesting is, remember that veins don't have the same amount of muscle layer, don't have the interior and external elastic membranes. But if a vein is asked to be an artery, and especially an artery like a coronary artery where there's a lot of pressure, it will remodel and start to look like an elastic artery. All right. And so that's why people who have bypassed, if you give them long enough, or if you let them continue to have their terrible diets and lifestyle and uncontrolled blood pressure, they'll get to a place where even their bypass piece of vessel that they got from a vein starts to be occluded like an artery and has to have bypass or stenting put in. Okay. And I do not really go other than that more on the vein. So I'll ask you artery arm, vein arms, artery leg, vein leg. I'll ask you artery abdominal or artery thoracic, but other than the major veins going to the heart, I won't be all over that. I'll ask you a little bit about the neck, head, and coming off the heart, arteries and veins. So make sure you know those arms and legs and the arteries for the trunk for the chest. All right, so are there any questions that have come up? Do we feel better about this? All right, so other than your ADI labs, now your ADI labs are going to be a little bit of this and a little bit of blood pressure and fluid flow velocity and a little bit of lecture material. And the reason why I kept those ADI labs is because I think they help, because blood pressure and fluid and resistance, and that's some tough physics. So I kept those materials in the ADI labs to help you with lecture material. So next week, there is no new material for lab. Okay, so next week's lab is all review. Okay, so if and when we do this on Thursday, it may be a 10-minute, we check in, we see if anybody has any questions, and that's it, because there's no more material. So those of you that come to the virtual labs, if you want me to go over any models, anything, review anything, that'll be your opportunity to ask. All right, the ADI LT labs, you have 2 due this week for week 4. Okay, and they are still due, and then in 2 weeks is your test. And again, during testing week, we will not have any labs, we'll not have any virtual meetings on Thursday and Wednesday for sure. If anybody really needs to take a paper test, I can put something together for lecture, for lab. I don't think I have a paper test. I could put one together, but I would prefer everybody just be on the same playing field and take lecture and lab at home with the video monitoring. Okay? Would you say making flashcards with labeling is a good study technique? For some people it is. For me, it was more of writing everything out was great. So I didn't necessarily have to make flashcards. I could just make like old school spelling things where I drew it out. And a lot of times, whatever I made flashcards, I never looked at them because I learned it through the writing. For other people, flashcards are helpful. Now, there are flashcards you can get pre-made from the websites or from certain textbook resources, or even just like buy them on Amazon. So again, if you like flashcards, but you don't want to take the time to make them, you might be able to buy some. For reviewing of ADI. So in one of my Bio 207 classes, one student took the lab exam and she was like, half of that wasn't on the ADI labs. ADI labs are supplements to this lab manual. So this should be where you spend your time first. And the ADI labs are for the points, as well as they are to supplement this, to help reinforce this. Anything in ADI that is not in this lab manual is probably either maybe lecture material or it's not testable. So the 12 lead stuff on the lab, you know how in the EKG lab there were some questions on the V1, the V2? That part is part of 12 lead. We don't do 12 lead. We only did lead 2 readout. Okay? So there is some stuff in ADI labs that is not necessarily testable. When you are in doubt, go to the lab manual and do 90 to 95% of your lab tests is this. And I leave a little bit of opening for some of that ADI LT lab and some of the lecture material to come over. Okay, does that help? Okay, any other questions? As I squat, oh my God.
So for this, for like all things, basically just have to memorize it.
Yeah, try to memorize them. I'll try to put my arrows on the vessels where they were in the book or close to that point. And then when you see it, my arrows will be purple, will be black, will be blue, will be red. So make sure you also look at the question. Don't just label them like based upon like superior to inferior or proximal to distal, because I might swip around and be like, what's the purple arrow line vessel? And that's the one pointing to the most distal vessel. You see what I'm saying? Okay. So again, I'm not going to ask them to you on the picture, just reading down the picture. I may ask them in a random fashion. Okay. And I will try to point to them again because if I'm pointing to it in the armpit, it's the axillary. If I point to it in the tricep bicep area, it's the brachial artery. So some of where I point to it matters. So I do try to hold to pointing to it close to where the book was pointing to it.
Also, like, are there like these pictures? Like...
Those are the exact pictures on your lab test. Okay. The ones in your lab manual, which is why I say 90% of your lab exam comes from your lab manual. So those are the pictures from your book. So if you are able to identify the vessels in the lab manual on those pictures, you should be good. I'm not, the only ones that I could do, again, on a model are the ones on the heart, okay? Any of these vessels on the heart are fair game. And then that's why I went through here really well. The middle, the inside of the torso. All right, Taylor, Erica, when is the lab exam? They will open on a Monday at noon. So the Monday 9 o'clock lecture on February 22nd, we're going to go over the quiz that you take the Sunday night. They will open at noon. You will have until Friday, 22nd, 23rd, 24th, something like the 26th You'll have through Friday to take it. So you can decide which one you want to take first. So if you're really good at the lecture exams and you want to get that one out of the way and then spend the rest of the week studying for the lab, that's fine. My Bio 207, I'd say 95% of the class has not taken either exam, and they're due tomorrow at midnight. So I suspect that everybody's waiting till Friday night to take them, and you do need to give yourself an hour for each test. So I wouldn't start both tests at 11 o'clock, because then you have 50 minutes or 59 minutes to take 2 tests. Okay, and they do require monitoring. And like I was telling someone yesterday, if you're going to have a sheet of paper, you need to show me during the environment check, this is the sheet of paper. And then as you're like doodling, every now and then just show the paper. And then at the end of the test, show me the paper. So that way I know you didn't slip a cheat sheet and you were doodling on your cheat sheet of material. I'm trying to give you guys the benefit of the doubt. If we start having cheating issues, then everybody's coming to campus and everybody's taking an in-person test. I mean, that would be the punishment. So, and again, the time and effort you put into cheating, if you just put that into studying, it's amazing. Normally you guys are fine. It's my 207. I'm like, seriously, just put the effort into studying and you'd be fine. Okay. If you'd really rather do at least the lecture test in person, you are welcome to send me an email and come on Thursday. I mean, as it is, out of 12 people, I have 2 here. So there's plenty of room to social distance and take a test if you want to take a lecture test. And then I think I have paper copies of lecture tests or lab tests 2 and 3 and 4. So, I mean, I can potentially give you guys in-person tests if for anxiety, or if you are just one of those people that really doesn't trust your computer, you really want to take it in person. Just email me. Let me go double check what I have. And then on that 23rd, so it'd be the 25th, Thursday between 1 and 3, I'm happy to give a test to you. Okay. So you can email me if that's really what you want to do. But I'm only coming to campus once for the 1 to 2 people. So everybody's got to, you know, I'm not going to come to campus twice a week for 1 person and 1 person. Okay. All right. Any other questions? All right. I'm going to stop the recording.